And we are live. Hello, everyone, and welcome for another webinar from our Open Neuroscience seminar series. As you know, it's still part of the Worldwide Neuro Initiative, and in our case, our series is about open source and open science tools for neuroscience. Today, we are very pleased to receive Suhasa Kodandaramaiah. Sorry if I say that wrong. He's from the University of Minnesota, and he's working in the field of neural engineering and neurotechnology design. His research combines principles of precision machine design, micro and nano fabrication, optics, and robotics to engineer better interface with the brain. He's our guest today as he has kindly accepted our invitation to discuss open source technologies for imaging cortex-wide neural activity. And I guess that most of our audience is already aware of his craniotomies project that has already been published in 2018. So, hello, Suhasa. Thanks for accepting being with us today.
Thanks for having me.
Okay.
Thanks for the kind introduction. It's really exciting to talk to a worldwide audience. I think this is my first worldwide seminar and probably one of the first talks that will go on YouTube, I believe, on your YouTube channel. So I will leave behind quite a few links and ways to get in touch with us if you're interested in any of the technologies I'm going to talk about today. As Maxine said, we are at the University of Minnesota, and I head the biosensing and biorobotics laboratory, where we're working on a number of different technologies for really large-scale interfacing with the brain. And why do we want to do that? One sort of a fundamental research question that we are kind of asking in our lab is, how do we navigate around the world, right? And these are pictures that I'm showing in and around the campus at the University of Minnesota. The picture on the left is a sculpture in front of the mechanical engineering building. That's my home. There's this interesting stairway in one of the buildings that I teach a class on machine design and mechanical engineering design.
Slides. Sorry for interrupting.
Oh.
We do not see your slides moving.
How about now?
No. Still the title. Sorry. We are stuck on a title.
Interesting.
Better. Okay, never mind.
How about now?
Yeah, now it works.
Yeah, so the picture on the left is a sculpture in front of my building. I use that as a landmark to navigate that when I'm outside, right? There are other features that might be around, right? Other sculptures that tell me I'm in front of the molecular and cell biology department, for example, right? But we're using multiple senses, right? We're using vision, we're using our sense of touch, our sense of how different textures of the landscapes and the ground that we navigate in. We might be using olfactory cues to do these navigations, and all of this is integrated in multiple regions, and this information is stored as memories and is flexibly used in different kinds of strategies that we're using to navigate the world, right? And if you think about it, a lot of this sensory information is coming to various parts of the cortex. I'm showing two different brains, one of the human, one of the mouse model that we study in our group, right?
We still on slides. It's not moving, sorry.
For some reason,
we are still on your cafeteria slide.
How about now?
Yeah. Now I see the brains.
You see the brains, and you're able to see the transition, right?
Yeah.
Yeah, great. Sorry about that. And different parts of the cortex are, as we know, are implicated in different kinds of sensory motor behaviors. And so we have different cortical areas that have specialized in processing visual information or somatosensory information. Then there are parts of the brain that are involved in motor function, for instance, right? And other parts that are involved in executive function, top-down control, so on and so forth. And so while neuroscience has traditionally focused on individual brain regions, more and more there has been an interest in understanding how all these different regions are simultaneously working together to mediate behavior, for instance, navigating around the world. And that's something that we are really interested in, both from an engineering perspective as well as from a basic science perspective. And for the most part, I'm going to talk about a suite of technologies we've developed over the last few years that allow us to access, image, large parts of the cortex during free behavior. And so I'm going to talk about a robotic cranial microsurgery platform that allows us to perform really large craniotomies relatively easily, polymer skulls that allow chronic optical access for very long durations of time, some strategies that we're using to functionalize these polymer skulls so we can not only do calcium imaging, but also perform electrophysiological recording and stimulation at the same time. And some compatible miniaturized devices we've developed for cortex-wide imaging. And a little snapshot of what some of these technologies can give us in terms of new kinds of scientific avenues that they're opening up. And then finally, I'll talk a little bit about some of the work we've done in terms of technology dissemination. This is something we're passionate about. How do we get these tools into the hands of the neuroscientists so more people can do the science? And with that, I'll end my talk. Now, one of the great things that has been developed over the last 15 to 20 years has been the development of genetically encoded calcium indicators, which can sense the influx of calcium while we are influx of calcium when neurons are firing action potentials. And this is shown in this sort of classic video now from the 2013 Nature paper describing GCAM6, where as cells are firing action potentials that are shown in the red trace below, there are the calcium ions come in and they bind to the genetically encoded calcium indicator. In this case, it's GCAM6 that results in these really nice changes in fluorescent intensity that serve as a really nice proxy for neural activity, right? And there have been more recent developments where, especially in the last few years, we've had some really exciting developments in the form of genetically encoded voltage indicators that have really fast kinetics, that can provide near single spike or even subthreshold information about what's happening within the neurons intracellularly using optical signals as a way to deduce these signals. And complementarily, we have these really nice tools that allow us to very precisely stimulate or silence genetically defined populations of neurons using optogenetics. So optics is a really nice way to think about interfacing with the brain. And the challenges when we go from imaging relatively small regions to say a challenge where we want to image the entire cortex, right, are sort of somewhat, it's not just about imaging, but it's also about how do you access the really large parts of the tissue, how do we maintain optical access for long durations of time. What do we do in terms of the design of technologies that can allow really large scale recordings while not compromising the behavior? And so I'll be talking a little bit about each one of these. Now, the first challenge, if any of you have done surgeries on mice, is that these surgeries are hard because the mouse skulls are relatively thin. For those of you who are not familiar, it has the similar structure anatomically to a lobster shell. You need neuroscientists typically use tools adopted from dentistry, dental drills and dental burrs that slowly chip away very carefully parts of the skull in a very controlled surgical setting to remove the skull and replace that with a cranial window. We wanted to see if we can automate this procedure because it can be very challenging when you think about replacing 80% of the mouse skull, right? That's covering most of the dorsal surface of the brain. And through this, we adapted what is essentially a hobby CNC machine that's used by woodworking hobbies. It has an XYZ stage with a spindle that can be programmed with micrometer scale precision to follow specific trajectories. Now, the question is, how do we get these trajectories? In our case, we first incorporated a home-built stereotax made out of off-the-shelf optomechanical parts that you can buy from Thor Labs, and 3D printed nose cones and ear bars that have been machined in the shop. And this allows us to fix a mouse in the CNC platform. And we use a really low force contact sensor, again, modified from an off-the-shelf surface profiler, in this case, designed to apply very low contact force while sensing the delicate mouse skull. And the profiler profiles the surface of the skull at a few different locations that is shown in the top panel. And it determines the X, Y, and Z locations of these points, interpolates a milling path that a programmed CNC mill can follow to delicately and very precisely remove micrometer thin layers of the bone to remove the skull. And so I'll show you an example of how this robotic craniotomy system works. Here's the low force contact profiler. It's profiling the skull surface at a few points along a circular path. And we're trying to implant a circular cover glass in this case. I will fast forward a little bit to the point where it finishes this profiling. And then we co-register a mill that's controlled by a computer. In this case, the mill has been programmed to go 50 micrometers deeper than the points that were profiled by the surface profiler. I want to mention that all of this is done in a very controlled fashion. Everything is sterile, just like the way you would do a surgery on humans, right? The mouse has been given anesthetics, and we take really good care of the animals before and after the surgery. So we inspect the skull. If it's not thin enough, the mill goes for another run. This time it's going 60 micrometers deep until it hits what is called the soft portion of the underside of the bone, which is called the trabecula, at which time it's relatively easy to use a pair of surgical forceps to remove the bone. And so what we're automating here is the really difficult part that humans find extremely hard to do, which is doing these controlled, precise removal of micrometer pieces of bone tissue, right? A little bit about the surface profiler itself. It's designed to operate at a very low force regime. We're talking about a few gram force, tens of gram forces, and it's fairly accurate. And so here's a micro CT scan of a skull surface. Overlaid on top are these yellow dots that were profiled using the skull surface profiler. And it has really low measurement error. It has a higher error when we go further lateral, owing to the higher curvature of the skull surface. But the measurement error is within a few tens of micrometers, and that's good enough for us to use the CNC build, right? And with this, we've shown that we can do a number of different procedures. We can perform circular cranial window implants and do longitudinal calcium imaging through these windows. More importantly, for the purpose that we set out to do a few years back, it allows us to reliably program these really large craniotomies, right? And we can program in features such as the little divot that you see in the craniotomy that's allowing us to avoid some major blood vessels along the central sinus, central suture, midline suture. And throughout, even though we are doing such a large craniotomy, we never touch the underlying soft tissue, either the dura or the bone or the brain, which is really important for the integrity of the next step, which is the implantation of the cranial window. Now, when you look at this, one of the things you immediately realize is that this is not a flat surface. If you're doing a cover slip implant over a relatively small area, 3 to 4 millimeters, we can implant these glass cover slips. But as you scale or increase the size of these cover slips, it becomes increasingly hard because there's a limit to how much you can flatten the brain surface, right? And so we really need, when we start thinking about large scale interfaces, we really need curved surfaces. And to be able to achieve this, we use the same CNC mill that I just talked about. In this case, we profiled the skull of one adult male C57 black sex mouse. Now, one of the great things about laboratory mice that we use in neurosciences, most of them are predominantly derived from the C57 strain. And because they're genetically conserved, once they reach adulthood, the size and shape of the skull is pretty much conserved across, across mice. And so you need to do this profiling on one mouse, and you can create a digital profile of the skull surface and use that to design these polymer skull implants that can be 3D printed using desktop 3D printers. And we use, typical tools that are used in the laboratory. Later on, I'll show you a picture of how we assemble this. Relatively straightforward, using adhesives and so on, and you can build an implant that is curved, conforms to the surface of the dorsal, the brain, and it can be implanted for really long durations to do very stable and chronic imaging of neural activity. And so here's one example of one of these polymer skulls that gives us access to over 45 millimeters square of the cortical surface, 6 weeks after implantation, 36 weeks after implantation. And you can see that these implants stay on for very, very long durations of time without much neuroimmune response when we assess either GFAP or GFAP, which is a marker for astrocyte aggregation, or IBA1, which I'm not showing here, but we've done this in the paper, which is a marker for increased microglia activity, right? So these polymer skulls made out of simple 3D printed polymers, plastics, can be implanted for really, really long durations and allow us to image neural activity chronically. And so here's an example of one of these implants on a transgenic animal expressing GCAM6 and predominantly most excitatory neurons using a Thai1 promoter. And using macroscopic imaging devices, we can start looking at the macro scale calcium activity as an animal is head fixed, right? But it also allows us to do really high resolution imaging if we need to, using two-photon microscopy on head fixed animals. And so Now, within the same device, you can not only look at mesoscale calcium activity, but also zoom in on individual microcircuits in a random access fashion and look at single cell activities. And you can do this over long durations of time. Here's one example where we were able to, in a random access fashion, access several different regions separated millimeters apart and look at single cell activities. Single cell activities. And we and other groups have estimated that using these kinds of implants and two-photon and three-photon microscopy, we have access to the activities of over 1 million neurons, right? Now, because this is a polymer and not a glass cover slip, it gives us additional flexibility to do things that would be hard to do with glass cover slip implants. So for instance, you can simply punch a hole using a soldering iron, cover that using a sterile layer of quicksil while it's implanted, peel off the quicksil, introduce electrodes so we can do intracortical microstimulation, for instance. And here's some examples of how microstimulation can be very different depending on whether the animal is anesthetized or awake, right? And so, and you could imagine doing other things. It really becomes a very flexible platform to do not only wide field calcium imaging or high resolution two-photon calcium imaging, but also combining it with other modalities, for instance, deep brain optogenetic stimulation or deep brain electrical stimulation, and so on and so forth. The next question we asked was, how am I doing on time?
Fine, just perfect. You can keep on going.
Sorry, I think I was on mute.
I said you can keep on going.
So can the seashells be more than passive transparent barriers, right? Optical imaging is great, but it's limited by temporal resolution. And if we look at some of the traditional clinical and translational data we have, it's primarily electrophysiological, right? Are there ways in which we can combine these two modalities? And here's a collaboration with one of the groups in the electrical engineering department with Professor Sarah Swisher, who is an expert in inkjet printing flexible electronics. In this case, we use a conducting and transparent polymer called P.PSS that can be used using some specialized inkjet printers that Professor Swisher has in her group to pattern really thin and transparent layers of conducting electrodes on top of these polymers. And so one of the things I forgot to mention is that the transparent part of the implant is a polymer called PET, which is polyethylene terephthalate, a much thinner version of what you see every day in your lives in terms of these plastic water bottles, right? It's biocompatible and probably the most recycled plastic in the world. And it's a great substrate for printing electronic devices on. And so here's an example where we inkjet print these electrodes, then we encapsulate them with Parallel C. It involves a little bit of clean room fab processing, but then we can build these fully integrated devices that seamlessly allow us to not only do calcium imaging, but also perform simultaneous electrical recordings. And these electrodes remain stable with low impedances, which is what you want for surface field potential recordings or electrocorticography, with lots of working electrodes throughout the duration of implantation. Here's a picture of one of these implants chronically implanted. And again, you can sort of see, you can image through these electrodes, right? And what this allows us to do, for instance, is start looking at what is the response across the cortex for a sensory stimulus. How does the state of the brain, whether it's actively whisking versus sitting passively, or whether it's attending to a task, affect not only the electrocorticography signals, but also the extent of the calcium activation, right? And so again, you provide a viscous stimulus, you can start localizing the peak calcium response to different electrode sites, but also look at what this does to the electrophysiological signals. And this is an ongoing study, and we're beginning to see interesting not only correlates between calcium and electrophysiology, but also instances where they significantly diverge in their responses. And we think this is a function of brain state, right? And sort of gives a sense for the kind of complementary information that we can get with these kinds of multimodal interfaces. Now, this work was done using highly specialized inkjet printers that have been developed for printing semiconductor rings and so on, and also involves clean room fabrication. And in the spirit of open neuroscience, this will be hard to disseminate unless we have a dedicated fab facility or a company that's thinking of commercializing this. And so this started off as an undergrad project where we said, can we build really high performance neural recording devices without ever entering a clean room, right? Things that everyday neuroscience lab should be able to do using readily available laboratory tools or with some help from outside, but then that should be minimal. And so we've been exploring open source, do-it-yourself, flexible neural recording devices, in this case, a graphene ECOG array. And we use essentially a form of printing that's been used in many different applications. And so if we have a laser cutter, and most universities have laser cutters, we could pattern stencils that can be, we can create stencils that can be used to pattern drop cast inks that can be sintered, covered with a sacrificial layer that can expose parts of the electrode traces that we need to be in contact with the brain. And then we need a way to insulate these electrodes so that just the part of the electrodes that are exposed act as reliable electrical interfaces. And so this is how we do it. We use laser cutters that you see in the bottom left of our screen to pattern these stencils. We can get close to 200 micrometer minimum feature sizes. The stencil on the bottom is the stencil that's used to pattern the electrode traces. And then the holes that you see on the top are used to pattern a sacrificial layer of a chemical called pluronic that's used to temporarily cover the electrode pad areas, right? And then we spin coat it using a home-built a baking dish microprocessor control spinner to pattern a really thin layer of an insulating polymer called PDMS. And we wash this gently in hot water, and pluronic melts when under a slightly higher heat than room temperature to expose the electrodes. And to do the final insulation step, we built this relatively simple spin coater, right? It uses an Arduino to control the speed of a servo motor, and the servo motor holds a rotating platform in which you place your substrate, and you spin it at different speeds to achieve various thicknesses, the desired thickness. And again, really simple design. This was a senior design mechanical engineering project that one student accomplished in a semester. And it gives us the ability to spin coat thin layers of insulating polymers like PDMS that you can bake in a laboratory oven and get really high performance flexible electrocardiography electrodes. And again, here's one example where these ECOG devices can be implanted for very long durations with low impedances, with greater than 90% of the electrodes functioning. And the beauty of this approach is that if you need to change the layout, if you need to change the design of the electrodes, you can do it very readily using the CAD files, right? That we use for patterning these laser stencils. And the whole process for building from the scratch, including the synthesizing of the ink, takes 2 days. And so in 2 days, you can have a batch of very reliable ECOG devices that you can adapt for whatever scientific study you want. And so this is a project that we are beginning to wrap up and should be available for the rest of the community very soon. I'm going to switch gears back to imaging. So what do we do with really large scale neural optical access, right? We want to be able to image not only in head fixed settings, but also in freely moving behavior context. That's where the full complexity in the behavioral repertoire can be studied, and potentially look at how multiple brain regions mediate these kinds of complex behaviors, right? And so we've developed miniaturized optical imaging devices that are compatible with the polymer sculls I just talked about. In this case, again, relatively simple optics. We're bootstrapping off of the open source mini-scope pipeline for a lot of our processes. So you have a lot of the design elements of the device. We have an array of 3 blue LEDs that are filtered, that are used for illuminating the GCAM. We have 1 green LED for reflectance. And so this is for mesoscale imaging, where we're not looking at single cell activity. And so you need this additional green LED for performing reflectance imaging, so you can identify and correct for hemodynamic changes. We have 1 collector lens and an emission filter and a top housing that includes CMOS sensor that's used as the imaging element. And this is a picture of the overall device. You can sort of see how a mouse is comfortable carrying this around in an open field arena. And this device allows us to image most of the field of view that we get through the polymer sculls, with the caveat that we are able to resolve features within about 30 to 60 micrometers. And so what this allows us to do is image with high resolution, but image a very large field of view, but at the mesoscale, right? So we're not able to distinguish individual neural activities, but we can, at the mesoscale, look at what's happening in terms of, is the retrospinal cortex co-activated with some of the frontal regions, so on and so forth, right? And we are beginning to see some really interesting things just with these kinds of capabilities. This is data showing how uniform the illumination is using the 3 LEDs, and this took quite a bit of optimization to get right. And these devices weigh roughly 3.8 to 3.9 grams. Once the animals get acclimatized, they're fairly comfortable. They can do a whole range of behaviors such as rearing and exploration. A little later, I'll show you a spatial navigation task that we have going while doing imaging, while doing mesoscale imaging. And they're sensitive enough to pick out very specific responses to sensory stimulus. And so in this case, a mouse that's anesthetized is provided a vibrational stimulus to the right forelimb, and you see this nice contralateral response in the in the somatosensory area, and the same mouse, if you provide a left visual blinking stimulus to the left eye, you sort of see a corresponding response to the contralateral visual cortex area, right? And so it's sensitive enough to pick out these sensory stimulation evoked activities. And we've been using these kinds of technologies to sort of look at how multiple cortical regions are being flexibly used during spatial navigation and learning. And so here's a behavioral arena, which is like the classic Barnes maze, where a mouse is introduced into this brightly lit and somewhat unfamiliar area where it has to learn to navigate to one of these holes, which has a safe hiding spot, and the rest are dummy holes, right? And while we're doing this, we are imaging the behavior of the animal, and we are imaging the whole cortex. And these experiments are done on type 1 GCAM 6F mice. And so here's some examples of the kinds of natural behaviors that we can get, right? And so mice introduced at the center of the maze will initially not know where the goal is. That's indicated by the green spot. But over time, and they take this random approach, but over time, within a few trials, they have these visual cues on the walls that they use to learn the schema of the search strategy, meaning the hole is somewhere at the edge, to memorizing the actual location. And so these are different search strategies. And one question, and as mice progress, you can sort of see that they have more higher percentage of the spatial search where they're directly navigating to the goal, and the latency decreases, and so on and so forth. And we're asking the question, what kind of spatial information is encoded in the cortex, right? And so this has been something that has been of increasing interest in the field. So here's some work from Bruce McNaughton's group, originally from the hippocampal place field studies Hall of Fame, right? And what they've shown is that a significant fraction of neurons distributed in many different cortical regions have spatial information. And so some of the questions that we are beginning to ask is, how widespread is the distribution of spatial information? Is it discernible in the mesoscale calcium activity? And is there something interesting in the emergent mesoscale cortical activity that encodes space, right? So just to give you a flavor of some of these questions, I'll give you a sneak peek of some of the things that we see, right? And so we started looking at mice that are using very similar paths to reach a goal. In this case, here's a mouse on 2 different trials that roughly follows the exact same path to get to the goal. If you compare the paths along the paths of the cortical activity when they're traversing the exact same locations, that's highlighted in sort of the bolder dots at the bottom left of your screen. We tend to start seeing that these mesoscale activities are, and this is to remind you, across the whole cortex, you sort of see similar patterns, right? And another way to visualize this would be to look at pixel by pixel as a function of distance from the goal. And we're starting to see these structures where you see hotspots emerge over the same pixels at the same locations in time, locations in space. And you can sort of start doing trial to trial correlations of various cortical regions of interest. And I'm showing you just snapshots of a few of these regions. When we sort of do these cross-correlation analysis, you sort of see that trial to trial, there's extremely high correlation between and significantly high correlations between a large fraction of the cortex. And so as mice are navigating these spatial arenas, there might be similar cortical activity at similar locations. And so we're beginning to explore this in greater detail. But I just wanted to give a sneak peek into the kinds of science we can start doing with these miniaturized, really wide field optical imaging devices. I wanted to end my talk with a couple of notes, right? One of the things is, as a tool building lab, we can build the technologies, and we may be able to use these technologies within our lab or in collaboration with few close collaborators. But increasingly, in the spirit of open neuroscience, right? One of the questions we all want to ask is, how do we get these into the hands of the research community, to as many people as possible? Because that's when we have the maximum impact, right? And that needs to happen right at the inception, excuse me, of the device itself. So for instance, one of the things I tell the students in my group is probably the most impactful and successful neurotechnology is the humble patch pipette, right? Because anyone can take a glass capillary, use heating filament, and pull a pipette that can do incredible neuroscience. And it costs a few cents for each glass capillary. And so technologies that we develop need to be accessible. If they're not immediately accessible, we need to figure out how to get them into the hands of the research community. And we use multi-pronged approaches to do this. One thing we've done diligently with all the technologies so far that have come out is we follow the original research publications with really detailed protocols where labs that are hands-on, who have the capabilities in terms of 3D printing and basic electronic processing, can follow these instructions. And we try to document them in as detailed fashion as possible so they can build these tools themselves. So here's an example of the automated craniotomy platform. Here's how we build these polymer skulls, right? Really simple 3D printed parts, glue, and a pair of scissors and a benchtop vise is all you want to build these polymer skulls. Or a little more involved where we have these assembly steps for building these miniaturized mesoscale imaging devices. So we do this diligently, and we found that a number of labs reach out to us saying, these are great instructions. I have a couple of questions about this particular step. And so we know that the threshold has been lowered somewhat when we do the documentation on our side. And a lot of this documentation will be available, and we update our GitHub every 3 months or so. And you can find them here. The second thing that we have done, actually this is the third, I think I got the numbers messed up. So this is the second thing we've done is we also worked with other stakeholders in the open source community, and so especially LabMaker. We spent about 6 months with the engineers at LabMaker building providing them with all the details they needed to build a turnkey version of the Craniobot. So if you're interested in having automated craniotomy capabilities in your lab, but you do not necessarily have either the time or the expertise to build one yourself and debug and test and so on, LabMaker has a turnkey version that they sell on their website. York Winter, who heads LabMaker, has told me they sold about half a dozen of these units, for instance, over the last couple of years. We also work really closely with a number of beta testing laboratories. We have provided seashell kits. We have provided mini-mScope kits, some of them which are fully built. So laboratories around the world can get these going. And we're beginning to see some of the fruits of this effort. It takes a significant amount of effort But once we do, it has a domino effect in terms of both in terms of the satisfaction we get in doing these kinds of open source activities, but also seeing how these beta testing laboratories go out and publish, and we start seeing other labs adopting the technologies that we've developed. And so if any of you are interested, definitely reach out to me either via Twitter or email. And also, all of this is provided on GitHub, and we try to open source pretty much everything. With that, I would like to end my talk. This is the team that we're working with, and our funding sources and collaborators. Thank you.
Thank you, Suhasa. That was really interesting. Actually, we do a lot of work like this in our lab, and I know how difficult it is to do craniotomy and see-through shell and electrophysiology on top of that with Windows. So really appreciate this kind of work. Really appreciate it. I'd just like to say to the audience that if you have questions, you can ask them directly to the chat, or I see there's many people with us right here. So if you want to turn on your camera and ask questions yourself, you are more welcome to do so. So I'll give you a couple of minutes just to join us. I'm actually impressed that it doesn't decay. I mean, after hundreds of days, it's still pretty clear. I'm very impressed with that. You are muted.
We have two types of animals. I think this is probably true for other, what do you say, cover slip implants too, which is we have a small fraction of animals that have some kind of clouding or whitening that happen. Usually, I would say about 10 to 15% of the animals. And it usually has something to do with the surgical procedure, because there are some surgeons in our group that have never had this issue. But once they go past the first 3 weeks or so, and if they remain clear, they can remain clear for a very long duration of time. And in the paper, we've shown data going up to over a year, right? So 370 days, where we can start tracking neurons over time.
That is impressive.
Yeah.
And our calcium marker doesn't decay or bleach, stay there.
We did a little bit of work on that. I think there's a little bit of signal to noise ratio decrease when we look at the single cells. But I don't know if anyone's looked at what the GCAM signal is 300 days or 360 days after implantation. So it was in response to a reviewer comment. We did the experiment. We saw a slight decrease. But this is for one strain of mouse. Who knows, how GCAM is replenished. Yeah.
Actually, when you said that, okay, you 3D printed with a resin-based printer, so you print your seashell. That you use a soldering iron to poke holes into it. How do you plug those holes after you finish your experiment?
That's a great question. I'll pull that up. So we implant, we put the implant. After there's a little hole, we cover it up with sterile silicone polymer called Quixel, and that seals it up. And whenever we need to access it during an experiment, we peel it off. The one caveat here is you want to take extra care of the animal and check on the silicone seal and make sure it's sealed and there's no, infection. So we do that a little more periodically, once a week or so.
That's definitely impressive. I know a lot of people that can benefit from that. So I know I'm going to share your talk around. Sure.
Cool.
Okay. So people from the audience, if you have, hello, Mikhail.
You can ask a question.
Hey,
very cool talk, first of all. And
thanks.
I had a few questions about the recording while they were kind of running around in the Barnes maze. So first off, I noticed you kind of displayed the trials on top of each other and looked at the correlation. How did you kind of line up the trials, since presumably there's kind of different amounts of time before they reached a goal on each one? Did you just kind of scale it linearly?
We are spatially averaging for doing this comparison, in the sense that, one, in this really preliminary analysis, in all of this, we are spatially averaging. And then if we are still not able to align them exactly, we just equalize the traces, the length of the traces, by adding zeros on one and on the other, right? And that's how we're doing the correlations. Now, I have to note that, so they are highly correlated. One of the questions that we still don't know is whether it's because they're in the same location in space, or whether it's something else, in the sense that, is it a state of the cortex? Meaning, are they in a particular attentive state, or whether they're using a particular strategy where they're looking for specific parts of the scene. There's more to this that we're excited about, but we still haven't done the full analysis. What I think we're seeing is that there are distinct states that that we see in the cortical activity, that are associated with a state of attention, a state of uncertainty, a state of having memorized where they have been before. And what you're seeing is a collection of all of this. And so we're seeing these correlations because it's not only that they're traversing the same locations in space, but they're also potentially having the same level of attention or using the same sort of serial search strategies and so on.
A kind of related question. You mentioned that there's these different strategies they use, the random, the serial, and the more directed strategies. Can you actually decode that from the neural activity in any way? And if so, kind of which regions seem to be important?
Yes. We've done a little bit. I do not have, I don't know if you're seeing this, we can decode the locations using a convolutional neural network. So we can train a model using just the snapshots of cortical activity and ask the model to predict where it is in space. And it's pretty good at doing that. Now, again, I don't have an answer as to why it's so good yet. Or I think I know, I just don't know if I can confidently talk about it yet. And what regions are important. I can tell you that there are specific patterns of activity that we see when an animal is reached a goal and doing exploratory behavior in and around a goal. That is very distinct from exploratory activities away. And we think both some of the retrospinal cortical regions as well as the frontal regions need to be simultaneously co-activated for this kind of exploratory behavior in the goal.
Great, thank you.
Definitely insightful. Thank you for that. Do we have anyone that would like to intervene? Tofi, hello.
Hi, can you hear me?
Yeah.
Yeah, thanks for a very nice talk. I had a question about so you said the seashell is made by or with a polymer. And are there any specific properties this polymer has to have to avoid any sort of optical aberrations, or the printer has to be of a certain quality so it's smooth enough, or that's sort of what's curious
polymer
about.
film we get is from DuPont. It's commercially available, 50 micrometers thick. They sell it by the commercial name of Melinex. Now, one of the things, we did some characterization of the polymer films. We showed, I don't think I have the data here in my talk. Let me see if it's a hidden slide somewhere. No, but in the paper, we do point spread function analysis as compared to a glass cover slip. Because these films are thinner than the usual cover slips that most optical objectives, imaging objectives are designed for, which is 170 micrometers or so, you will see some decrease in the optical imaging performance. But we're able to see structures as small as dendrites when we do structural imaging on Taiwan GFP mice. So that's one. The second thing is, as compared to glass, these polymers absorb some of the blue wavelengths, and there's a little bit of autofluorescence. And so when we do two-photon imaging, stacked imaging, you can sort of see a thin layer of the polymer coming up in your two-photon Z stacks. And so there's about 3 to 5% greater loss in terms of the power of the light that can be transmitted through these polymers as compared to glass cover slips.
Yeah. And is this actually printed, or because now I'm looking at your slide, so that you also have a mold? Is the mold printed and then?
The mold is printed. The black frame that you see is printed. And then we take the polymer film that's cut, and then we use the molds to sort of conform and glue it to the frame. And so this provides a structure, and the polymer, the film provides a transparency.
And maybe if I may ask one last question related on that. So how difficult is it then to attach the seashell onto the skull? I mean, I guess you use dental cement or do you use something else? And especially like on the sides, I guess you go a bit far off to the sides with the seashell.
I want to see if I have a good.
What are sort of the tricky parts related to that?
The tricky parts are making sure, and we talk a lot about this in the protocols paper. You're making a large craniotomy, and so you need to be quick once the bone is removed between when you place the implant on the skull and seal it up. So that's the trickiest part, right? And so you want to limit the amount of time the brain is exposed, limit the amount of time the dura starts drying out. But then you want to keep the brain moist, but you want to keep the skull as dry as possible. So the implant sticks. Yeah, that's the tricky part. And generally what we've noticed is the people who are more practiced, who do these surgeries faster, have really good success rates. Okay. And so I have a student who can finish the surgery in an hour and 45 minutes, and she is the master surgeon, right? And there are others who take a lot longer.
Okay, thanks.
Thank you.
Good technical question. Thanks, everyone. Do we have a last one question? 3, 2, 1. Okay, then. So, thanks again for the talk. Thanks again to our audience to be there. I hope we answer most of your questions. That's definitely an interesting project, and we will keep looking into it. And we will definitely look at any upgrade you will have in your system and your under review papers.
Thank you.
For sure.
Thank you so much for hosting me.
Thank you, everyone. Stick with me for a minute. I will just end the live stream.